The Biology of Belief, Chapter 1, Lessons from the Petri Dish, in praise of smart cells and smart students. On my second day in the Caribbean, I stood in front of more than 100 visibly on-edge medical students. I suddenly realized not everyone viewed the island as a laid-back refuge. For these nervous students, Montserrat was not a peaceful escape, but a last-ditch chance to realize their dreams of becoming doctors. My class was geographically homogeneous, mostly American students from the East Coast, all races and ages, including a 67-year-old retiree who was anxious to do more with his life. Their backgrounds were varied. Former school teachers, accountants, musicians, a nun, a drug smuggler. Despite all the differences, the students shared two characteristics. One, they failed to succeed in the highly competitive selection process that comprised American medical schools. Two, they were strivers, intent on becoming doctors. They were not about to be denied the opportunity to prove their qualifications. Most had spent their life savings or indentured themselves to cover tuition and extra costs of living out of the country. Many found themselves alone for the first time in their lives, leaving families, friends, and loved ones. They put up with the most intolerable living conditions on campus, yet with all the drawbacks and the odds stacked against them, they were never deterred from their quest for a medical degree. Well, at least that was true up to the time of our first class together. Prior to my arrival, the students had 3 different histology cell biology professors. The first lecturer left the students in the lurch when he responded to some personal issue by bolting from the island 3 weeks in the semester. In short order, the school found a suitable replacement who tried to pick up the pieces. Unfortunately, he bailed 3 weeks later because he got sick. For the preceding 2 weeks, a faculty member responsible for another field of study had been reading chapters out of a textbook to the class. This obviously bored the students to death. But the school was fulfilling a directive to provide a specified number of lecture hours for the course. Academic prerequisites set by American medical examiners have to be met in order for the school's graduates to practice in the U.S. For the 4th time that semester, the weary students listened to a new professor. I briefed them on my background, my expectations. I made it clear, even though we were in a foreign country, I was not going to expect less from them than was expected by my Wisconsin students. Nor should they want me to, because to be certified, all doctors have to pass the same medical boards, no matter where they go to medical school. Then I pulled a sheaf of exams out of my briefcase and told them I was giving them self-assessment quiz. The middle of the semester just passed. I expected them to be familiar with half of the required course material. The test I handed out consisted of 20 questions from my own histology midterm exam from Wisconsin. The classroom was deadly silent for the first 10 minutes. Then nervous fidgeting filled the students, one by one, faster than the spread of the deadly Ebola virus. By the time 20 minutes allotted for the quiz were over, wide-eyed panic gripped the class. When I said stop, the pent-up nervous anxiety erupted into excited conversations. I quieted the class down, began to read them the answers. The first 5 or 6 answers were met with subdued sighs. After I reached the 10th question, each subsequent answer was followed by agonizing groans. The highest score in the class was 10 correct answers, followed by several students who answered 7 with guesswork. Most of the rest scored at least 1 or 2 correct answers. When I looked up at the class, I was greeted with frozen faces. The strivers found themselves behind the big 8 ball. With more than half a semester behind them, they had to start the course all over again. A dark gloom overcame the students, most of whom were already treading water in their other demanding classes. Within moments, their gloom turned into quiet despair. In profound silence, I looked out over the students. They looked back at me. I experienced an internal ache. The class collectively resembled one of the Greenpeace pictures of baby seals before fur traders clubbed them to death. My heart wailed. Perhaps the salt air and sweet scents made me more magnanimous. In any case, unexpectedly, I found myself announcing I'd make it my personal commitment to see that every student was fully prepared for the final exam, if they would commit to providing matching efforts. When they realized I was truly committed to their success, I could see the lights flash on in their previously panicked eyes. Feeling like an embattled coach revving up the team for the big game, I told them I thought they were every bit as intelligent as the students I taught in the States. I told them I believed their stateside peers were simply more proficient at rote memorization, the quality that enabled them to score better in medical college admissions tests. I tried to convince them that histology and cell biology are not intellectually difficult courses. I explained that in all of its elegance, nature employs very simple operating principles. Rather than just memorizing facts and figures, I promised they were going to gain understanding of cells because I'd present simple principles on top of simple principles. I offered to provide additional night lectures, which would tax their stamina after their already long lecture lab-packed days. The students were pumped up after my 10-minute pep talk. When the period ended, they bolted from the classroom, snorting fire, determined they would not be beaten by the system. After the students left, the enormity of the commitment I had made sank in. I started having doubts. I knew a significant number of the students were truly unqualified to be attending medical school. Many others, capable students whose backgrounds had not prepared them for the challenge. I was afraid my island would degenerate into a frenetic, time-consuming academic scrimmage that would end in failure. I started thinking about my job at Wisconsin, and suddenly it was beginning to look easy. At Wisconsin, I gave only 8 lectures out of the approximately 50 that made up the histology cell biology course. There were 5 members of the department that shared the lecture load. Of course, I was responsible for the material because I was involved in the lab sessions. I was supposed to be available to answer the course-related questions. Knowing the material and presenting lectures on the material are not the same thing. I had a 3-day weekend to wrestle with the situation I created for myself. Had I faced a crisis such as this back home, my type A personality would have had me swinging from the proverbial chandeliers. Interestingly, watching the sunset in the Caribbean, the potential angst simply morphed into an exciting adventure. I began to get excited about the fact that for the first time in my teaching career, I was solely responsible for this major course, free from having to conform to the style and constant restrictions of team-taught programs. Cells as miniature humans. As it turned out, this histology course was the most exhilarating, intellectually profound period of my academic career. Free to teach the course the way I wanted to teach it, I ventured into a new way of covering the material, an approach that had been rolling in my brain several years. I'd been fascinated by the idea that considering cells as miniature humans would make it easier to understand their physiology and behavior. As I contemplated a new structure for the course, I got excited. The idea of overlapping cell and human biology rekindled the inspiration for science I felt as a child. I still experience that enthusiasm in my research lab, though not when I was mired in the administrative details of being a tenured faculty member, including endless meetings and what, for me, tortuous faculty parties. I was prone to thinking of cells as human-like because after years behind a microscope, I'd become humbled by the complexity and power of what at first appeared to be anatomically simple moving blobs in a petri dish. In school, you may have learned the basic components of a cell: the nucleus that contains genetic material, the energy-producing mitochondria, the protective membrane at the outside rim, the cytoplasm in between. Within these anatomically simple-looking cells is a complex world. These smart cells employ technologies that scientists have yet to fully fathom. The notion of cells as miniature humans that I was mulling over would be considered heresy by most biologists. Trying to explain the nature of anything not human by relating it to human behavior is called anthropomorphism. True, scientists consider anthropomorphism to be something of a mortal sin and ostracize scientists who knowingly employ it in their work. However, I believe that I was breaking out of orthodoxy for a good reason. Biologists try to gain scientific understanding by observing nature, conjuring up a hypothesis of how things work. Then, they design experiments to test their ideas. By necessity, deriving the hypothesis and designing the experiments require them to think how a cell or another living organism carries out its life. Applying these human solutions, for example, a human view of resolving biology's mysteries, automatically makes these scientists guilty of anthropomorphizing. No matter how you cut it, biological science is based to some degree on humanizing the subject matter. Actually, I believe the unwritten ban on anthropomorphism is an outmoded remnant of the Dark Ages, when religious authorities denied direct relationship existed between humans and any of God's creations. Well, I can see the value of the concept when people try to anthropomorphize a light bulb, a radio, a pocket knife. I do not see it as a valid criticism when applied to living organisms. Human beings are multicellular organisms. We must inherently share basic behavioral patterns with our own cells. However, I know it takes a shift in perception to acknowledge that parallel. Historically, our Judeo-Christian beliefs have led us to think we're the intelligent creatures who are created in a separate, distinct process from all other plants and animals. This view has us looking down our noses at lesser creatures as non-intelligent life forms, especially those organisms on the lower evolutionary rungs of life. Nothing could be further from the truth. When we observe other humans as individual entities or see ourselves in the mirror as an individual organism, in one sense, we're correct, at least from the perspective of our level of observation. However, if I brought you down to the size of an individual cell so you could see your body from that perspective, it would offer a whole new view of the world. When you look back at yourself from the perspective, you would not see yourself as a single entity. You would see yourself as a bustling community of more than 50 trillion cells. As I toyed with these ideas for my histology class, the picture kept recurring in my mind was a chart from an encyclopedia I used as a child. Under the section on humans, there was an illustration with 7 transparent plastic pages printed with identical overlapping outline of the body. On the first page, outline image of a naked man. Turning the page was peeling his skin, revealing his muscles. On the second page, the images remained a dissection of the body. Flipping through, I could see in turn the skeleton, the brain, the nerves, the blood vessels, the organ systems. For my Caribbean course, I mentally updated those transparencies with several additional overlapping pages, each illustrated with cellular structures. Most of the cell structures referred to as organelles, miniature organs within cytoplasm. Organelles are the functional equivalent of tissues and organs of our bodies. They include nucleus, the mitochondria, the Golgi body, vacuoles. The traditional way of teaching the course is to deal first with these cellular structures, then move to the tissues and organs of the human body. Instead, I integrated the 2 parts of the course to reflect the overlapping nature of humans and cells. I taught my students that the biochemical mechanisms employed by cellular organelle systems are essentially the same mechanisms employed by our human organ systems. Even though humans are made up of trillions of cells, I stress there is not one new function in our bodies that is not already expressed in a single cell. Virtually every nucleus-containing cell possesses the functional equivalent of our nervous system: digestion, respiration, excretory system, endocrine, muscle, skeletal, circulatory, integument, skin, reproductive system, even a primitive immune system, which utilizes a family of antibody-like proteins. I also made it clear to my students each cell is an intelligent being that can survive on its own, as scientists demonstrate when they remove individual cells from the body and grow them in a culture. As I knew intuitively when I was a child, these smart cells are imbued with intent and purpose. They actively seek environments that support their survival while simultaneously avoiding toxic or hostile ones. Like humans, single cells analyze thousands of stimuli from the microenvironment they inhabit. Through the analysis of this data, cells select appropriate behavioral responses to ensure their survival. Single cells also are capable of learning through these environmental experiences and are able to create cellular memories which they pass on to their offspring. For example, when a measles virus infects a child, an immature immune cell is called in to create a protective protein antibody against the virus. In the process, the cell must create a new gene to serve as a blueprint in manufacturing the measles antibody protein. The first step in generating a specific measles antibody gene occurs in the nuclei of the immature immune cell. Among the genes and a very large number of DNA that encode the snippets of protein. By randomly assembling and reassembling the DNA segments, immune cells create a vast array of different genes, providing a uniquely shaped antibody protein. When an immature immune cell produces an antibody protein that's a close physical complement to the invading measles virus, the cell is activated. Activated cells employ an amazing mechanism called affinity maturation that enables the cell to perfectly adjust the final shape of its antibody protein so it'll become a perfect complement to the invading measles virus. Using a process called somatic hypermutation, activated immune cells make hundreds of copies of the original antibody gene. However, each new version is slightly mutated, so it'll encode a slightly different shaped antibody protein. The cell selects the variant gene that makes the best fitting antibody. This selected version of the gene goes through repeated rounds of somatic hypermutation to further sculpt the shape of the antibody to become a perfect physical complement of the measles virus. When the sculpted antibody locks on the virus, it activates the invader and marks it for destruction, thus protecting the child from the ravages of measles. The cells retain the genetic memory of the antibody, so in the future the individual is again exposed to measles, the cells can immediately launch a protective immune response. The new antibody gene can also be passed to all the cell's progeny when it divides. In this process, not only did the cell learn about the measles virus, it also created a memory propagated by daughter cells. This amazing feat of genetic engineering is profoundly important because it represents an inherent intelligence mechanism by which cells evolve. The origins of life. Smart cells get smarter. It shouldn't be surprising cells are so smart. Single-cell organisms were the first life forms on the planet. Fossil evidence reveals they were here 600 million years after the Earth was formed. For the next 2.75 billion years of the Earth's history, only free-living single-cell organisms populated the world. Amoeba-like protozoans, bacteria, algae. These smart cells figured out how to get smarter when first multicellular organisms appeared. Cellular communities consist of tens of hundreds of cells. The evolutionary advantage of living in a community soon led to organizations comprised of millions and trillions of cells, each cell of microscopic dimensions. Biologists classified these organized communities based on their structure. Cellular communities appear as single entities to the naked eye, a mouse, a dog, a human. They are in fact highly organized associations of millions and trillions of cells. The evolutionary push for bigger communities is a reflection of the biological imperative to survive. The more awareness an organism has of its environment, the better its chances for survival. When cells band together, they increase their awareness exponentially. If each cell were to be arbitrarily assigned an awareness value, each colonial organism would collectively have a potential awareness value of at least X times the number of cells in the colony. In order to survive at high densities, the cells create structured environments. The sophisticated communities subdivide the workload with more precision and effectiveness than the ever-changing organizational charts that are a fact of life in big corporations. It proved more efficient for the community to have individual cells assigned to specialized tasks. In the development of animals and plants, cells begin to acquire these specialized functions in embryo, a process of cytological specialization enabling them to form specific tissues and organs. Over time, this pattern of differentiation, the distribution of the workload among the members of community, became embedded in the genes of every cell in the community, significantly increasing the organism's efficiency and its ability to survive. So each cell developing genes and other cells to cooperatively promote the survival of the whole body of cells. Differentiation of a person in a family or community or group is each person becoming differentiated and developing into an emotional, physical, psychological, spiritual being that promotes the survival of all the other beings as well as that own person's self. Amazing. In larger organisms, for example, only a small percentage of cells are concerned with reading and responding to an environmental stimuli. That's the role of groups of specialized cells that form the tissues and organs of the nervous system. The function of the nervous system is to perceive the environment and coordinate the behavior of all the other cells in the vast cellular community. And I say that part of differentiation because that's David Schnarch's work with his book, Brain Talk, that talks about people becoming differentiated in families and creating happier families through that process. Going on. Division of labor among the cells in the community offered an additional survival advantage. The efficiency it offered enabled more cells to live on less. Consider the old adage, 2 can live as cheaply as 1, or consider the construction costs of building a 2-bedroom single home versus the cost of building a 2-bedroom apartment in a 100-apartment complex. To survive, each cell is required to expend a certain amount of energy. The amount of energy conserved by an individual living in a community contributes to both an increased survival advantage and a better quality of life. In American capitalism, Henry Ford saw the tactical advantage in the differentiated form of communal effort and employed it in creating his assembly line system of manufacturing. Before Ford, a small team of multi-skilled workers would require a week or two to build a single automobile. Ford organized his shop so every worker was responsible for only one specialized job. He stationed a large number of these differentiated workers along a single row, the assembly line, passing the developing cars from one specialist to the next. The efficiency of job specialization enabled Ford to produce a new automobile in 90 minutes rather than weeks. Unfortunately, we conveniently forgot about the cooperation necessary for evolution when Charles Darwin emphasized a radically different theory about the emergence of life. He concluded 150 years ago that living organisms are perpetually embroiled in a struggle for existence. For Darwin, struggle and violence are part of animal and human nature and the principal forces behind evolutionary advancement. In the final chapter of The Origin of Species, Darwin wrote of the inevitable struggle for life, that evolution was driven by war of nature, famine, and death. Couple that with Darwin's notion that evolution is random, and you have a world described by Tennyson characterized as red in tooth and claw, a series of meaningless, bloody battles for survival. Evolution without the bloody claws. Though Darwin's by far the most famous evolutionist, the first scientist to establish evolution as a scientific fact was a distinguished French biologist, Jean-Baptiste Lamarck. The leading architecture of neo-Darwinism, a modernization of Darwin's theory, incorporates molecular genetics, concedes Lamarck was the pioneer. The book Evolution and Diversity of Life, Mayer wrote, "It seems to me Lamarck has a much better claim to be designated as the founder of theory of evolution, as indeed he's by several French historians. He was the first author to devote a book primarily to the presentation of a theory of organic evolution, the first to present the entire system of animals as a product of evolution. Not only did Lamarck present his theory 50 years before Darwin, he offered a much less harsh theory of the mechanisms of evolution. Lamarck's theory suggested evolution was based on instructive, cooperative interaction among organisms, and that environment enables life forms to survive and evolve in a dynamic world. His notion was that organisms acquire and pass on adaptations necessary for survival in a changing environment. Interestingly, Lamarck's hypothesis conformed to modern cell biologists' understanding of how cell immune systems adapt to environment. Lamarck's theory was an early target of the church. The notion that humans evolved from lower life forms was denounced as heresy. Lamarck was scorned by his fellow scientists, who as creationists ridiculed his theories. A German developmental biologist, August Weismann, helped propel Lamarck into obscurity when he tried to test Lamarck's theory that organisms pass on survival-oriented traits acquired through their interaction with the environment. In one of Weismann's experiments, he got off the tails of male and female mice and mated them. Weismann argued if Lamarck's theory were correct, the parents should pass on their tailless state to future generations. The first generation of mice was born with tails. Weismann repeated the experiment for 21 more generations, but not one tailless mouse was born, leading Weismann to conclude Lamarck's notion of inheritance was wrong. But Weismann's experiment was not a true test of Lamarck's theory. Lamarck suggested evolutionary changes could take immense periods of time. According to biographer Jordanova, in 1984, Jordanova wrote Lamarck's theory rested on a number of propositions, including the laws governing living things have produced increasingly complex forms over immense periods of time. Weismann's 5-year experiment was clearly not long enough to test the theory. An even more fundamental flaw in his experiment is that Lamarck never argued that every change an organism experienced would take hold. Lamarck said organisms hang on to traits, like tails, when they need them to survive. Although Weismann didn't think the mice needed their tails, no one asked the mice if they thought their tails were necessary. Despite its obvious flaws, the study of the tailless mice helped destroy Lamarck's reputation. In fact, Lamarck had been mostly ignored or vilified. One wrote in The Evolution of an Evolutionist, Lamarck's the only major figure in the history of biology whose name's become, to all intents and purposes, a term of abuse. Most scientists' contributions are fated to be outgrown, but a very few authors have written works which, 2 centuries later, are still rejected with indignation so intense that the skeptic may suspect something akin to an uneasy conscience. In point of fact, Lamarck has been somewhat unfairly judged. Waddington wrote those words 35 years ago. Today, Lamarck's theories are being reevaluated under the weight of a body of new science that suggests the oft-denounced biologist was not entirely wrong and the oft-lauded Darwin was not entirely correct. The title of an article in the prestigious Journal of Science was One Sign, Was Lamarck Just a Little Bit Right? One reason some scientists are taking another look at Lamarck is that evolutionists are reminding us of the invaluable role cooperation plays in sustaining life in the biosphere. Scientists have long noted symbiotic relationships in nature. And Darwin's blind spot, British physician Frank Ryan chronicles a number of such relationships, including a yellow shrimp that gathers food while its partner protects it from predators, and a species of hermit crab carries a pink anemone on top of its shell. Fish and octopus like to feed on hermit crabs, but when they approach this species, the anemone shoots out its colored tentacles with their microscopic batteries of poison darts, stinging the potential predator, encouraging it to look elsewhere. The warrior anemone gets something out of the relationship as well because it eats the crab's leftover food. Today's understanding of cooperation in nature goes much deeper than the easily observable relationships. Biologists are becoming increasingly aware that animals have co-evolved and continue to coexist with diverse assemblages of microorganisms required for normal health and development. A new article in Science called We Get By with a Little Help from Our Little Friends. The study of these relationships is now a rapidly growing field called systems of biology. Ironically, in recent decades, we've been taught to wage war against microorganisms with everything from antimicrobial soap to antibiotics. But that simplistic message ignores the fact that many bacteria are essential to our health. The classic example of how humans get help from microorganisms is the bacteria in our digestive system, which are essential to our survival. The bacteria in our stomach and intestinal tract help digest food and also enable the absorption of life-sustaining vitamins. This microbe-human cooperation is the reason the rampant use of antibiotics is detrimental to our survival. Antibiotics are indiscriminate killers. They kill bacteria required for our survival as efficiently as they kill harmful bacteria. Recent advances in genome science have revealed an additional mechanism of cooperation among species. Living organisms, it turns out, actually integrate their cellular communities by sharing their genes. It had been thought that genes are passed on only to the progeny of an individual organism through reproduction. Now, scientists realize genes are shared not only among the individual members of a species, but also among members of different species. The sharing of genetic information via gene transfer speeds up evolution since genes can acquire learned experiences from other organisms. Given this sharing of genes, organisms can no longer be seen as disconnected entities. There's no wall between species. The manager of Energy's microbial genome program told Science, "We can no longer comfortably say what is a species anymore." This sharing of information is not accident. It's nature's method of enhancing the survival of the biosphere. As discussed earlier, genes are physical memories of an organism's learned experiences. The recently recognized exchange of genes among individuals disperse those memories, thereby influencing the survival of all organisms that make up the community of life. Now that we're aware of this intra-species gene transfer, the dangers of genetic engineering become apparent. For example, tinkering with the genes of a tomato may not stop at that tomato, but could alter the entire biosphere in ways that we cannot foresee. Already, there's a study that shows that when humans digest genetically modified foods, the artificially created genes transfer into and alter the character of the beneficial bacteria in the intestine. Similarly, gene transfer among genetically engineered agricultural crops and surrounding native species has given rise to highly resistant species deemed superweeds. Genetic engineers have never taken the reality of gene transfer into consideration when they've introduced genetically modified organisms into the environment. We're now beginning to experience the dire consequences of this oversight as their engineered genes are spreading among and altering other organisms in the environment. Genetic evolutionists warn that if we fail to apply the lessons of our shared genetic destiny, which should be teaching us the importance of cooperation among all species, we threaten human existence. We need to move beyond Darwin theory, which stresses the importance of individuals, to one that stresses the importance of the community. British scientist Lipton provides evidence that evolution is more dependent on the interaction among species than the interaction of individuals within a species. Evolution becomes a matter of the survival of the fittest groups rather than the survival of the fittest individuals. In an article in Nature, Lipton wrote, "Rather than focusing on individuals and their role in evolution, we must consider the totality of organisms and the material environment to fully understand which traits come to persist and dominate." Lipton subscribes to Gaia's hypothesis that holds that Earth and species constitute one interactive living organism. Those who endorse this hypothesis argue tampering with the balance of the superorganism called Gaia, whether it be destroying the rainforest, depleting the ozone layer, altering organisms through genetic engineering, can threaten its survival and ours. Recent studies by Britain's Natural Environment Research support those concerns. While there have been 5 mass extinctions in the history of our planet, they're all presumed to have been caused by extraterrestrial events, such as comet smashing to Earth. One of the new studies conclude that the natural world is experiencing the 6th major extinction event in history. This time, the cause of the extinction is not extraterrestrial. According to the study's authors, Thomas, as far as we can tell, this one is caused by one animal organism, man. Walking the talk of cells. In my years of teaching in medical school, I'd come to realize medical students in an academic setting are more competitive and backbiting than a truckload of lawyers. They live out the Darwinian struggle in their quest to be one of the fittest, who stagger after graduation after 4 years in medical school. The single-minded pursuit of stellar medical school grades without regard for the students surrounding you no doubt follows a Darwinian model, but it always seemed to me an ironic pursuit for those who are striving to become compassionate healers. But my stereotypes about medical students toppled during my stay on the island. After my call to arms, my class of misfits stopped acting like conventional medical students They dropped their survival of the fittest mentality and amalgamated it into a single force, a team that helped them survive the semester. The stronger students helped the weaker. In doing so, they all became stronger. Their harmony was surprising and beautiful. In the end, there was a bonus, a Happywood Hollywood ending. For their final exam, I gave my students exactly the same test the students in Wisconsin had to pass. There was virtually no difference in the performance of these rejects and their elitist counterparts in the States. Many students later reported when they went home and met with their peers who attended medical schools, they proudly found themselves more proficient in their understanding of the principles governing the life of cells and organisms. I was, of course, thrilled my students had pulled off an academic miracle, but it was years before I understood how they were able to do it. At the time, I thought the format of the course was key. I still believe the overlapping human and cell biology is a better way to present the course material, but now that I've ventured into what I told you would be considered by some a wacky Dr. Dolittle territory, I think a good part of the reason my students succeeded was that they eschewed the behavior of their counterparts in the U.S. Instead of mirroring smart American medical students, they mirrored the behavior of smart cells, banding together to become even smarter. I didn't tell my students to pattern their lives after the lives of the cells because I was steeped in traditional scientific training. But I like to think they went in that direction intuitively after listening to my praise of cells' ability to group together cooperatively to form more complex, highly successful organisms. I didn't know it at the time, but I now believe another reason for my students' success was that I did not stop at praising cells. I praised the students as well. They needed to hear they were first-rate students in order to believe they could perform as first-rate students. As I'll detail in future chapters, so many of us are leading limited lives not because we have to, but because we think we have to. But I'm getting ahead of myself. Suffice it to say, after 4 months in paradise, teaching in a way that clarified my thinking about cells and the lessons they provide to humans, I was well on my way to an understanding of the new biology, which leaves in the dust the defeatism of genetic parental programming, as well as survival of the fittest, Darwinism. And that concludes Chapter 1.